Okay, so it's 8:30. So what I recommend is we'll get people are starting to hop on here. So we'll get off to a good start. So I just want to welcome all the attendees and panelists. My name is Danielle Reed, and I'm on the leadership team of the GCCR, and I'm also your technical host today. So if you're having questions or concerns, you can drop your messages to me in the chat. And if you want, I'll be monitoring both the chat and the Q&A and be helping Dr. Hoover out, Kara Hoover is going to be our chair for today. And I'm going to turn it all over to her. Okay, go ahead, Kara.
Hi, I'm the chair of the GCCR DEI committee, and one of our committee members, Marius Baguma, organized this webinar, but unfortunately he wasn't able to chair it today because he's off to Belgium to defend his dissertation. So I have stepped in his place, and we can all wish Marius good luck today or over the next few days as he goes through the process. But I am delighted, though, to be able to introduce our first webinar hosted by the Diversity, Equity, and Inclusivity Committee. Our goal is to promote work that either fosters DEI or is conducted by non-majority researchers or in marginalized research areas or parts of the world. So our remit is quite broad, and this might include you even if you don't think it does. So if you are actually interested in participating in our DEI webinar series, please do contact us. And maybe at the end, I'll put up the slide with our email address. So I do want to thank Marius again for organizing this. And we have 3 speakers today who are either former, current, or incoming members of the DEI committee. So I'll briefly introduce each speaker, who will have about 10 to 12 minutes of floor time. And then this will give us a little time to switch between speakers and still have about 15 to 20 minutes at the end for questions and answers. So as we go, just feel free to put any questions or comments you have in the Q&A, and we can address those at the end. So, it can be in any order for any speaker. So I'm going to start off with Ritesh Kumar. He received his PhD from the Academy of Scientific and Innovative Research in India in engineering science, and he is currently a principal scientist at the Central Scientific Instruments Organization in Chandigarh, India. And I will give the floor to you, Ritesh.
Thank you, Karen. Can I share my screen now?
You may, go ahead.
Sure. Is it visible?
Yes. It is. So you just need to go full screen.
Yeah. So is it full screen now?
Yes.
Yeah, and then I can move it.
You're full screen and you're moving great, and we can hear you fine.
Thank you. Thank you very much. First of all, a big thank you to GCCR, Kara, Marius, and all the DI members, and of course the GCCI leadership team and everybody there to give me an opportunity to speak on some of the recent and some earlier work that I have been doing in this field, especially olfactory research. This is the work that we have been doing as a group since some time, and this presentation will not always necessarily be completely my work, but yes, this is a work of a group also. In the end, I'll also show who are part of this group. So yes, as told, I'm principal scientist at Central Scientific Instruments Organization. So what we have placed ourselves is in the area of olfaction and taste. We do work with perfume and ether industries. That's one of the oldest industries still surviving, with lots of artisans here in India in terms of this called as micro, small and medium enterprise. One of the companies there is Flavor and Fragrance Development Corporation in Kannauj. So we do a lot of work with them in aroma profiling. We work in food and AI. We work on the technology part also, that is developing electronic nose and tongue systems. And on top of it, big data and machine learning, all of these works. That does not mean that they do not overlap with each other. They are just for the purpose of showing that they are independent, but yes, they are not only independent. So the fundamental question that we try to solve is, why does a molecule smell as it smells? So in this, what we have done is we have taken an objective step of identifying the physical space, relating it to the perceptual space at this moment. For that, what we have done is we have identified lots and lots of data, extracted lots and lots of features from this data. This is the data from perfumery, agriculture, odor experiments, and research papers of the molecules, and through some series of experiments of data processing, data cleaning, we relate it to the perceptual, to the words that are told by the speakers, by these researchers, or even by experts in this. So we have published all of these things in PLOS One and other journals. Even as a team, we were also part of one of the olfaction consortium that identified that the paper came in Science. So we were able to identify the features, and more than half of the number of times in experimentation, we were able to show that you can predict the perceptual descriptor of a molecule using the machine learning techniques. So, and beyond that, what we have done is we have tried to use some deep learning techniques, some state-of-art deep learning and machine learning techniques to identify the nature of the fragrance space. And so what you see here as a picture is a simple embedded representation of the molecules, in which the distinctive molecules are are part of the fragrance space. They lie on a certain axis. This is the principal component analysis of the embedding space. We showed this result in ECRO Tuesday. I think it should be 2019. If I'm not wrong, 2020 is wrong. So yeah, so we showed this result in 2019 that yes, embedding representation are one of the most powerful representation which could be used for understanding the fragrance space. There we introduced also the concept of molecular fragments from this, where we designed an algorithm in which we showed that how some of the fragments of molecules, again through machine learning techniques, and identifying clear which of the part of the molecules are clearly responsible for a particular smell. So and we showed this result in ISOT 2020. Later on, one of the very interesting things that we also did was designing a molecule. Say, for example, if you start with a seed molecule, can you use generative models, for example, in deep learning to generate new fragrant molecules, which are not necessarily of the same odor family type, but which can be of which can have a certain odor. So we were able to in fact generate some of the molecules. Of course, there were problems in terms of the molecules not being thermodynamically possible, but yes, some of the results were really encouraging, and we showed this result again in ECRO Dresden 2020 there. So moving on to we have been working on developing of electronic tongue instrument, which can differentiate between different different basic tastes. So we developed an electrical impedance spectroscopy based technique to clearly identify between different tastes, and we also showed that this tongue was able to differentiate between tea of different origins and quality and water of different origins and quality, using again machine learning and other techniques, and we published it in Sensor and Actuators P, and it was and what it showed was that even a two electrode setup could be useful for this kind of experiments and for this kind of instrument design. So and as I said, we are very closely working with micro, small, and medium enterprise, such as which are there in ether making, perfume making processes. So this is like one of the very old industries that has been still going on in the Kannauj, in the northern part of India. So what we did was, without changing the technology part of it, in the sense of the work that they were doing, we were trying to understand what goes on into that old process by using simple process monitoring techniques, and we set up this system there in different at different places, at different artisan homes, and we were able to show some interesting things about essential oil mixture processes, such that Like goes with like. Some interesting smells go together and some do not go. And you could use it for designing perhaps essential oil mixtures. There again, moving on to one of the very interesting works, we also worked with cashew nut industries in trying to identify the volatiles which were responsible for the rancidity of this. What was happening is these cashews were getting rejected because they turned out rancid at some point of time, and they were not able to understand why is it happening. So we identified the processes and the volatiles which were responsible for this using gas chromatography and electronic nose technique to identify the culprit volatile, if I may, and to clearly mark out the instrument which can be used to find out that. Moving on to one of the very interesting work on taste analysis, we work with one of the biggest consumer companies, which is Tata Consumer Drugs Limited. So this was on green tea bitterness masking and saltiness differentiation, objectively trying to understand the masking agent that they used. Were they able to clearly differentiate between were they able to reduce the bitterness or not, and quantify it? And also the modeling taste profiles of tea oleoresin, elaichi flavor, if we call it, with respect to time, as to say, for example, if the oleoresin is kept for a certain period of time, what changes it? What change in flavor takes place from there? And one more work, very interesting, very recent work that one of my colleague and me, we have done is we have developed an algorithm for food recommendation. This is also with a particular company, in which we designed a non-Euclidean embedding of the descriptors in recommending what food would you like based upon, for example, the Netflix. We see that movies, as a movie recommender or book recommender system, they have certain characteristics. Now they depend upon big matrix, in which sparse matrix, where you want to fill out it's basically a matrix factorization technique. But we showed that it's if you take out, for example, if you take food, and if you have somehow the information about the olfactory characteristic of these food items, then you could use it, and you use it to design an algorithm which will be better than the existing algorithms in recommending the food items. So what was done? We designed a non-Euclidean embedding of the descriptors by defining a graph between the descriptor from the earlier knowledge that we had, that I showed you in the first slide, and we transferred that embedding knowledge of the percept to the attribute space, and that's where we got the product embedding. Now, is this the same? So yeah. So one of the more interesting work that one of the PhD, one of the master students that I worked with, we worked on the odor source localization, and we published on IEEE transaction in cybernetics in 2018 and 19, is trying to understand how multiple robots can reach a certain source of an odor without any other information, for example, without any visual information, just following the plume trail. This is a problem which has been explored a lot of times in literature, but yes, we showed that our algorithm, in fact, worked better than most of them at that time, and to reach the global maximum. One of the things that we assumed was around the source, there will be a global maxima, and there will be lots of local maxima in and around the source. And finally, one of the things that I did when I came to GCCR, when I participated in GCCR, is I was one of the persons conducting the home test part of it in India. We designed the questionnaire, we designed the algorithms, and in fact, this questionnaire was I would say was developed with the help of the GCCR leadership and the persons who were actually involved in the home test at that time. But it was a bit different than the global GCCR this thing home test, because there are some of the spices and other things were different in India. So we designed the algorithms, and we developed the models, and this work is still in the under review. Future, lots of things that I want to do we want to do as a group in short term, lots of things, plant health monitoring, machine learning for mobility solution, other kinds of things. There are lots of odor source localization and lots of things that we want to do. And this is the team that I'm part of. These are the funders that we have. I've been very lucky to be funded by the Royal Society as a part of Newton Fellowship. CSIR, which has funded me in my development in these last 5-6 years. Tata Consumer Products Limited and FFDC and Nanopix. Thank you very much.
Thank you, Ritesh. That was a fascinating collection of very diverse and eclectic research. So we're moving now to Jingguo. And if anybody has any questions, please feel free to put them in the Q&A. We'll address those at the end. We're moving now to Jingguo Chen, whose PhD institution was Xi'an Jiaotong, not sure if I said that correctly, University Health Science Center in China, and also 1 year spent at the University of Colorado in the Anschutz Medical Campus as a visiting scholar. His current position is the Department of Otolaryngology, a head and neck surgeon, second affiliated hospital of Zhangjiaktong University, and a research fellow and also scientific research secretary. His primary discipline is pharmacology, and he'll be speaking to us today about his views of chemosensory research in China. So I will give the floor to you.
Okay, thank you very much. Hello, everybody. It's my great honor to be here. It's a valuable opportunity for me to give a presentation on the GCCR DEI webinar. So today I will give a short presentation about the title. It's the chemosensory science in China from my personal views. I'm working in the Department of Otolaryngology, Head and Neck Surgery in the second affiliated hospital of Xi'an Jiaotong University in China. So, okay. The following, I will following the 4 aspects to introduce my presentation. First, I want to give us a brief self-introduction. I was a graduate in Xi'an Jiaotong University in the 2018, major in pharmacology, and as a visiting scholar, studying the University of Colorado, Denver, Anschutz Medical Campus, supervised by Susie Kahneman. And for now, after I got my PhD degree, and then I worked in the Department of ENT in the university, and also published some papers about like a beta T receptor in the human sinonasal cavity, and also the beta T receptor in arteries. It's showing some extraoral physiology function of the beta T receptor. Also recently, I cooperated with the Richard Doty in the University of Pennsylvania, and doing some the self-administrated water lines empirical test and test WETT in the China population. Also, it's honor to join the become a GCCR member, also as a member as a co-author in the past the chemosensory journal, published 2 articles. As you know, he's shown he is there on the world map. There are only 4 members from China, the GCCR member. It's only a few of patients, including the scientists or doctors, so focusing on the chemosensory, it's smell or taste research. So it needs to do a lot of work to raise awareness from them. So I want to talk about why I meet with GCCR. It's very early of the pandemic. It's 2020, February 27. At that night, I was want to go to bed and looking at my phone to notice some news reported that COVID-19 patients have anosmia as the only symptoms. So I got up very quickly to sit down on the desk to search on the websites to review the published papers and recently the new reports. Then I wrote and submitted an article about the COVID-19 and the olfactory disorder, maybe as the early symptoms of the COVID-19. Submit the short paper to the JAMA Otolaryngology, but it's rejected on the March 1st of the 2020. And then I don't know why, but maybe it's my poor English or my poor writing English. Then I transfer my direction to conduct some of the popular science works warned the public should concern the olfactory symptoms during the endemic by social media, such as my personal social media, also including my some our university's official website to show awareness of the patients, to show need to concern the loss of smell and the COVID-19. After that, it's in about the March 20, Clary Hopkins, the president of the British Rhinological Society, it's just released a media statement about the COVID-19 and reports the smell loss as a symptom. So after that, I noticed that, oh, there's a great group. So it's called GCCR, and then I become members doing some following jobs. Also to raise the idea to why we cannot set up for a World Taste and Smell Day. So last year, under the guidance of Daniel Reid and many staffs from worldwide to do some the first activities on the World Taste and Smell Day. Also, we're doing some activities in China. It's only one the university or one hospital to doing activities on that day. We're also doing some like the free olfactory test to patients, also doing some like a popular science to the patients. It's also my great honor to I have interviewed by the China Global Television on the Global Watch. It's to introduce our the GCCR work, our GCCR the first paper, the first data to the audience. Also got some broadly awareness of that. So on the second part, I want to give a brief the science research progress in China. But now I searched on the website of the Web of Science. It's shown that in the title, the smell is about 64,000 publications from it's all this is from China. It's maybe ranked 10 about that. That's also most of these papers just focusing on like the first the computer science and engineering, also the neuroscience. About the taste in China, it's also ranked. It's about 7 on or 8 eighths. As the taste research is focusing on the food science, technology, and neuroscience, neurology, biochemistry, molecular. So it's also some many people are focusing on the scientific research, but they don't have the awareness to broaden the knowledge of the taste and smell to the public. So also under the GCCR work, we also apply for use the existing the data study, doing some curious disease on the change of taste and smell in the COVID-19 patients. Also doing like some the arteries and the nasal cavity. Also, it's not young. This is for my task or my previous scientific research. It's just very common. It's recently in China. It's also a very high quality. As a high quality paper is just published in PNAS about the olfactory regulation by a dolphin and the DRD2 receptor in the nose. It's very interesting. also is in my universities, Professor Chun-Shun Wu is also focused on the field effect sensors using biomaterials for chemical sensor in China. So it's many people on the different fields to do some chemosensory research on that. For the clinical field, it's also some people, some scientists also doing some like self-reported meaning of factory questionnaire for it's very easier and useful measurement for screening the factory dysfunction. But for now, in the clinical evaluation, the methods in China for the patients is very limited. Which is in my clinic, it's also used like the sniff sticks or upsets. It's fortunately in recently, it's also China scientists also developed the Chinese smell identification test. It's just it's there some cultural difference, so it changed some. It's similar with the sniff sticks. In clinical, we also need to use the MRI of the olfactory system to find how about the histology of the olfactory bulb. For the taste testing, it's just we don't have the commercial product. It's just using cooperate with Professor Richard Doughty to use a waterless empirical taste and test, WTT. So let's just give maybe a short few minutes to talk about the future directions and collaborations. We want to, it's a great opportunity to have this worldwide webinar to show what we have do, what we have done in the worldwide. So like we need two collaborations to do more tasks to raise the awareness of the public people. And also it's like recently we just talked about that. It's maybe following the tasks, for example, just the taste and smell clinical world map or the GCCR many lab study. It's maybe in the future, we also want to organize so like the Chinese Chemosensory Association. That will be better for raise awareness in China for most of the people. That's all. Thank you. Thank you very much.
Thank you very much. That was very interesting. A nice overview of what we can see coming out of China and maybe what we can expect to see in the future. Our final presentation is from Renee Hartig, who received her PhD from the International Max Planck Research Center for Neural and Behavioral Sciences in Tübingen University, Germany. She's currently a postdoctoral fellow with a focus program for translational neurosciences at Johannes Gutenberg University in Mainz, and her discipline is translational neurosciences. I will now give the floor to her.
Okay, thank you so much. So I'm just going to go ahead and share my screen, and we will start from the top. So yes, today I'm going to talk about translational and transnational chemosensory science. And the research component of this work, I just want to acknowledge from the get-go the work done in Tübingen with my mentor, Henry Edward, and in the Department of Nikos Logothetis. And the work that I do also in Germany. So pretty much all of my research right now is in Germany, but I'm also working with the Systems Neuroscience and Mental Health Group. That's a part of Heidelberg University and Central Institute for Mental Health. And my mentor, Wolfgang Kelsch, who's also at the University of Mainz. So just an introduction, chemical senses, taste and smell, they can be used to study neural circuits and information processing in health, helping to better understand basic neural mechanisms and how such mechanisms may be altered in different states of disease. Preclinical work in rodents and non-human primates may bridge the gap in understanding brain disease and in translating knowledge to sustain healthy brain function in humans. With a model sensory system and organism in place, single cell population activity and systems level correlation of neural activity are investigated in brain regions responsible for processing chemosensory inputs. And one of the regions that I have a particular focus on happens to also be the insular cortex, which I'll highlight particularly in my first case example. So what I'm mostly focusing on is studying and addressing functional homology in the primate brain. These efforts serve to validate and better inform the employment of non-human primate models in neuroscientific research. So what I can do, for example, with in a non-invasive means is with an animal model, the macaque monkey, for example, is put the animal inside the scanner. And as you can see here, we have an MRI image. And from that, we can have then a parcellation of different anatomical regions from atlases. And while we're scanning the animal, we can introduce different paradigms. So here we have a taste paradigm, and we have different tastes that we present to the animal in the scanner. And we were able to work with 9 macaques in this case with a fairly balanced male to female ratio. So from these data, what we can do at the group level is try to localize the primary area for taste processing in the cortex. So if we look at taste, just taste as compared to artificial saliva or a tasteless solution, some of the areas that we're localizing are actually within the insular cortex itself. And that actually includes bilaterally a localization in the mid-dorsal insular cortex, as well as ventral to that in the disc granular insula. And if you go a little bit anterior, you have another area that's also incorporating a special cell type. It's called the von Economo neuron. And this is a cell type which might then provide a top-down back to autonomic pre-brainstem nuclei that you can maybe get an idea of an ascending sensory circuit, and also then how the cortex is playing a role in processing this information. And with an invasive means, with a lower count, we can go in and try to then record from these cells in particular where we've localized from our search volumes by fMRI. And so to kind of put that into a perspective of other research that we know, for example, with the intra-insular activations, is that they may be selectively influenced also by the ongoing physiological state, such as satiety, anticipation, expectation. And what's interesting now is when you take the literature together with humans, macaques, and rodents. So rodents is where I would bridge to next as kind of just an example of what we can do with the animal models. So what can be accomplished in lower order mammalian species in regards to understanding neurocircuitry? Well, we can study chemosensory circuits in mice. It's an economical way to infer population effects and bridge insight on circuit conservation across species. So here, this is the work that we've now done in the mice. We have 2 interacting mice. And while the mice are interacting, we can record from the brain. We can also perturb circuits. And what we actually have done is we put different mice in boxes that allowed the odors of the mice to be relayed to an animal in the scanner. And here you can see is the animal in the scanner. The animal is awake. He's patiently waiting for the odors to arrive. And as that's happening, we're recording what's going on in the brain. Now, in another setting, we could outside the scanner also do electrophysiology. And we could actually do that in each mice after they've gone through their scan period. And while that's occurring, we can look at some physiological readouts to help us infer the arousal or saliency of these stimuli. And that includes pupil dilation, as well as the sniff response. So one of the questions we asked here was, how do social stimuli and social experiences influence odor processing? So in this first experiment, what we tried to do was distinguish odor types in the state of passive odor processing, where the mouse is in the scanner and the mouse is receiving social odors and non-social odors. The non-social odors here being peanut butter and a floral odor, and the social odors in this case being a conspecific of the same age or a juvenile or another adult, but from a different strain of mice. So what we looked at was the odor stimulus classification and decoding by fMRI and physiological readouts. So what we can show here, for example, is a differential testing of correlated bold responses to 2 different stimuli that we presented in the scanner. in this case, the peanut butter and the floral. And they do disassociate, and this was helped, the inference was helped by actually looking at the sniff cycle. So if you could see, for example, once the peanut butter smell comes, the amplitude of the sniff is heightened. And once the young comes, now while the young doesn't necessarily bear any aversive trait to it, it is a higher concentration, and it is a mask that goes fully over the nose. So in this case, we can see that the mouse is reacting to the high concentration, and the mouse will then pause during that sniff cycle. And so this really helps us to infer, taking both the physiology and what's going on in the brain, to try to get an idea of maybe how the mouse is perceiving the stimulus. And so what we can see here is that these appetitive social odors, that they drive bold activity in the anterior piriform cortex, which is, I could show you here if you follow my mouse right here, the olfactory tubercle, which is just ventral to that, as well as the olfactory bulb, which is anterior. And if we were to go back and look at our insular cortex, you can also see the agranular, which corresponds to anterior insular cortex in primates, lighting up for the peanut butter. So we wanted to take this a step further and see if we could modulate the social experience. And this, in this case, we can maybe do by oxytocin. So as inferred by previous reports, oxytocin is a neuropeptide, and in the social realm, it affects perception, and it can alter behavior and also influence the body physiology. So what we have found is that oxytocin administered during social interaction with the same species conspecific, it results in increased exploration events. So in this case, we have injected a virus into the paraventricular nucleus of the hypothalamus. This is where you'll find these oxytocin neurons. And what we do is, using optogenetics, we perturb the circuitry to drive, in this case, the oxytocin release from these niche of cells during a social interaction. And so as compared to controls, those that received the oxytocin explored more and for longer. This paves the road for oxytocin to be seen as a potential booster for social interactions. So we then went on to just do a couple more experiments with the social interaction to consider state-dependent processing, arousal, exploration. And we did that while we triggered the oxytocin release during the social interaction as a replication of Otto et al., which was done also in the lab of Wolfgang Kelsch. And we then also triggered the oxytocin release during scanning without social interaction. And so what we find here was, first, we confirmed by histology that indeed we did have our oxytocin neurons labeled. And we then looked at what then happens if we are giving the oxytocin during the social interaction and we put the mouse in the scanner. We can then actually see that the bold response disassociates between whether that animal was familiar or unfamiliar. So that means here is a familiar animal that this animal in the scanner is sniffing and getting to know, and we're contrasting that to another animal who is a stranger. And we can actually see that the response of the familiarity disassociates. And so just comparing that now to experiment 3 without the social interaction, but still driving oxytocin release, you can see now where you have the overlap. And I've kind of tried to highlight it here. We actually find that the overlap is in a cholinergic circuit hub, the diagonal band of Broca in this case. But then we can also notice anteriorly that we do have differential responses in the olfactory bulb that cross between a dorsal ventral extent. So overall, using this methodology, we can get a sense of where we might have overlapping anatomical regions playing a part in different types of paradigms. So attempts to bridge the understanding of chemosensory processing, it extends beyond translational study across humans and animals. Such attempts, consider also the transnational capacity to carry out and conduct such research. So this is where I will talk about the last part, and that really stems to Trend in Africa. It's a nonprofit organization currently based in the UK. There's a nice neuron paper which you could find some of the further information related to the effects of trend efforts. So those efforts, for example, include organizing courses. We have a lot of neuroscientists a part of the Trend Network. So we do have particular neuroscience courses, and we do try to get balance in inclusion of female and male participants, and basically building on those participants as our alumni to build our network across Africa. So one important point to consider here is the lack of local science infrastructure. It often leaves researchers more discouraged to pursue research, and it promotes a brain drain. One thing that we tried to highlight here is that there's a lack of access to modern equipment and new techniques significantly affecting the scientific progress in Africa. You can see the report in Nature Communications, and basically where they've tried to now quantify the number of papers coming out of different African countries. And that might be related to the fact that there isn't a lot of the tech that might be needed to produce those results. One of those pieces of equipment, for example, could be a confocal microscope. And there's only a few of them on the continent and only one in Nigeria. And I focus here in Nigeria because this is actually the birthplace of the Biomedical Science Research and Training Center. It's called the BioRTC. It's located and associated with the Yobe University, and that's in Nigeria. And it's been founded by Dr. Mahmoud Mena. He's also a part of Trend in Africa. And what you could do is you can navigate over to this site at any time. But the goal here of BioRTC is to build a local facility to train and support scientists to use what they've learned for their research. So one thing that we can note is we have available at the BioRTC not only a confocal, but also fluorescence imaging equipment. So this is actually now part of the network that we hope to share with other scientists in Africa, organize programs where participants can come and receive training. We're trying to also expand this network within Africa and also with partners who extend to countries outside of the continent. And there are also ways you might be interested to help, and they would be well received. So, I just wanted to mention that we're always looking for equipment and consumables to help build up the facilities and the capacity building efforts. And we're also looking for volunteers. And I know that I've spoken to, just by speaking, learning that others are interested in these opportunities. And honestly, I think a lot of the volunteer efforts really help the organization and the efforts to grow. So without further ado, I would like to thank you for your attention, and that would conclude my segment.
Thanks very much, Renee. Interesting hearing a little bit more about what you're doing as well as the Trends in Africa program. So we can open the floor to questions. We don't have anything in the Q&A right now, but if people have questions, they can put them in there or they can raise their hand. I just wanted to follow up actually with Ritesh and ask you a little bit more about the natural language programming that you did. I'm a little bit curious about that method. Did you essentially identify odors online and how people were describing them as a way to create your model for predicting perception?
Yes. Yes. As a part of natural language, the two aspects to it. First is of course from the molecule side, and second, of course, the perceptual descriptors. So in the first work that we did not use the natural language processing technique, some fancy natural language processing from the molecule side. We did all the work from the perceptual descriptor side. For example, let's say when you describe 5-6 words, and how do I get that information from? So, and how do I say that this word is similar to the other, and how do I remove the redundant information? There what we did was something called as stemming, but stemming did not always work because, say, for example, something like alcoholic and alcohol and the words they somehow started mixing up, and they did not give good. So we actually had to do manual manually we had to tokenize these words and find, sometimes, not always, these were all semi-automatic methods where we found that there was a problem. So, and then these molecules were described by, let's say, 5-6 words, sometimes more than that also. So in a sense, it became, if I say very simply, it became a multi-label, multi-class problem.
And did you
Which,
use where did you get the data from? Did you scrape it from the web, or did you
yeah,
specifically?
you scrape from the web, some of the database. For example, you have Sigma Aldrich database. We got it, we bought it from the PDF, and then we scraped the PDF and other kinds of things, and then we scraped from the web. That's one of the things, and from the literature as well. There also we scrape the literature to automatically scrape the literature to get the information of the molecule and the this thing and the perceptual descriptors.
Okay, great, thank you. We have a question also now in the Q&A for Ritesh. She wants to know, what are the machine learning models you used and what are their performances for odor prediction?
Yeah, so there was I would say, if you followed this line of research, you would have seen that in 2017 there was a big, I think, science paper. In fact, in 2017 it came in Science Advances, which where different kinds of machine learning techniques. For example, one of the very famous techniques and which were used and which performed the best was random forest there. In fact, other techniques also work very well. I have worked with random forest and other techniques. So it was like it's like a cocktail of different techniques that work together rather than a single technique performing the best, I would say. But yes, random forest, I would say that it sort of as a single modeling technique, it was at par with the other techniques as well. And there in the more than half of the times we were clearly able to predict what were the descriptors of the molecules. You could even reverse engineer it, but I would not say that that is the end of the road, because what we are doing here is we are taking there's a certain part of the whole chain where we are taking it as a black box, in the sense that input is the molecule and the output is the perceptual descriptor. Where is the other part? Where is the molecular interaction with the receptors and other things that are going on there? So that was not done at that time. But yes, in fact, in recent times, people have it's mainly because it's very clear in this community that it's a combinatorial explosion kind of problem. It's a many-to-many relationship between one set to the other, and we still do not know what are the receptors which will be activated clearly with these different molecules and the other way around. So that's the challenge, and yes. That's what it is. I think that's the long answer of your short question, I would say.
There's a follow-up question. Did you try neuron network? I think it must be like,
oh yeah,
neural
neural network. In fact, I tried, and many people, in fact, Google has tried. Google has published very recently something called as graph neural network on this, and they say that they have that works best than even better than what was published in 2017. So yes, I've also tried neural network techniques, but I have taken it from a different angle now. I'm not concentrated on predicting. I'm trying to explore the fragrance space in mathematical sense, because I think just predicting perceptual descriptors won't be enough. Again, because we are missing the link between the molecular and the receptors unless we have that complete data, or perhaps certain kind of data which we can completely rely on, in the sense of machine learning this thing. I'm looking mostly from the from the fragrance space in mathematical sense of this thing at this moment.
Thank you. I think we have somebody who's put their hand up. Marha?
Yes, thank you. I have a question. You can hear me normally? Okay. I have a question for Jingjuo Chen. And I was wondering, can you tell us a little bit more about the wet test, the taste test, waterless taste test, and how applicable it is in global settings or maybe remote settings or non-laboratory settings?
No, it's not. It's just need to taste some strips. Taste some the strips. It's just a powder on some like a sugar or nitric acid on the paper and to pack on the pad, and then to put them on the edge of the tongue and to test it. It's including it's including, yeah, it's 27 strips. It's $3 of the strips. Each of the box is including 9 strips.
And what are the strips made of? Is something that dissolves on the tongue, or is it something that you take off again?
No, it's disposal. I can show you some of the pictures of the WTT. Wait a moment. Can I share my screen?
You should be able to.
Okay. The paper is just accepted by the journal Sensory Studies, and it's cooperate with Richard Doty. Okay.
I was just looking for the paper. Congratulations on that.
Thank you very much. We have tested the so 120 people on Chinese people, and also 100 more than 100 in American people. Oh no, it's oh. Can you see that?
What we saw was a screen, a blue screen with word on it. So I don't know if that was your intention.
It's a Word file. It's
You're
okay?
welcome to, you can drop that in the chat too, just in the interest of time, that would be fine.
Yes, you can put a link to the paper if there's a version online to share.
Okay.
Thank you. And Danny, I think you had your hand up.
I'm going to push our panelists to answer a broader question. So in the last couple years, we've really seen two trends. We work locally and we communicate now better globally. And this is a fabulous example of that. So you're working locally, you're communicating globally. We've also seen through the GCCR that we're now able to work together, work globally with research partners. So my question to the panelists, and I'd like to hear from each panelist, is which is having the most powerful effect on your own research program? The being able to communicate globally, or are you working more with international partners, and is that driving change in your own research programs? So I'll just burn up 2 seconds and talk a little bit more, but I'd like to hear from Renee and Jingao and Ritesh on that question.
Okay, I can start. So I think probably a lot has been changed by the pandemic. I think how we can communicate virtually, it's exceeded maybe what we even thought could be possible. So for example, I've been invited to help join BioRTC as a researcher, but it's very clear that I wouldn't physically be in Nigeria all the time. I can come for a couple weeks here or there, but that's largely okay because the capacity to network with others on the ground, I feel it's more accepted. We maybe have had the tools of Zoom before, but it's really coming at secondhand. And so I think that's a little bit of just kind of being open to allowing the situation and normalizing that to occur, and not to particularly judge somebody because they cannot physically be there, but rather perhaps we can measure the success by the output. So having these clearly defined goals that you can define together with your international partners on a call guides pretty much the work efforts. And so I think it's like a little bit of a mix, a little bit of an acceptance that we don't have to physically be in one place in particular to work with our partners. And overall, I think we're getting pretty effective at this. I think we've learned over the past 2 years how to manage this. And so I feel like it's removed some of the barriers that geography, money to travel has maybe had in place even 5 years ago.
Yeah, I would agree with Renee on this, especially the two aspects to, of course, one is doing research, one is presenting your research in a conference and other places. So earlier, of course, you had to travel, and you need money for that. You need grants and other kinds of things. For a person, for example, who is the other, most of the conferences, let's say, I would say, quote unquote, big conferences, some conferences which are given some name, they happen in generally in America or Europe and other places. So it becomes quite difficult for a person, both monetarily, because sometimes you have grant, sometimes you do not have grant, and other kinds of things. So this has done wonders. I would say that now you do not have to go there. You can present there. Of course, there will be registration fees, but that can be managed somehow, because conversion rates, all those things, rates are still high. Flights are still costly. So that's what it is. So, and everything is driven by money. So that's what. So I would say that that's one of the things that I would say a big factor: presenting work. Journals are okay, conferences. Second, something like this, I don't think I would have thought, say 4 years back, that I would be presenting, or even 3 years back, I'd be presenting to some people of diverse countries at 6:30 p.m. in India, sorry, 7 p.m. in India. No way. Not possible. So this is something that has changed. How this has changed funding scenario, of course, you can do some research which does not involve funding. How this has helped funding scenario, I cannot tell at this moment, because funding is still similar. Governments, because that is controlled by governments, organizations. It's the same, but of course, technology has brought us together. Thank you.
Okay, from my points, it's as I'm a researcher in the clinical in the hospital, so maybe in the future we need to more collaboration in the worldwide. So doing some work on the diagnosis or the future the advanced treatment for the olfactory or gustatory disorders. But now like some like the stem cells or taste and smell regeneration, or we doing some the world clinical map, or many the GCCR many labs like those studies we also can doing some like our factory or taste test in the worldwide to find which is the difference between the two country the many countries or many like the ethics. So also maybe it's a science collaboration with also maybe like association, like the patient advocate associations doing some shared the popular science to the worldwide people and translated the same article or same popular science article to the worldwide to raise awareness in the worldwide to especially after the pandemic in the worldwide, because most of the people they should awareness. Oh, this is maybe the sudden loss of smell, loss of taste. We should care. Maybe it's just the COVID-19 infected or some others like the virus infected. So it's a lot of what we can do. It's just for a better life. Maybe it's benefits for everyone in the world. That's all. Okay, thank you very much.
Thank you. We have 1 minute left, and I want to take this as a opportunity to thank everyone for participating and attending today, and also plug this webinar series. So if you are interested in joining a future webinar, we aim to do this at least once a year, possibly twice a year. And if you are interested in putting your name in the hat to be considered, please do email us. And you can also use that email address to contact us in general. And I just realized there is a typo. There's no period after committee in the email address. So please do feel free to drop us a line. And again, thank everyone for attending and participating. And hopefully we'll see you at another webinar for the DEI soon. Thank you. Bye.
Thank you very much. Thank you. Thank you.
Thank you.
Bye,
Bye-bye.
everybody.
Bye.